Okay, so in this lecture we're going to discuss a couple of different topics. We are now into the special senses. So for this lecture what I'm going to talk about first off is smell, and then I'll also go over taste. And so if you find my lectures helpful, make sure to like and subscribe. So the first thing I want to point out to you, here I have the brain model. And the first thing I want to point out is the olfactory nerve. So that's cranial nerve number 1. So cranial nerve number 1 sits right here, and these are what's known as the olfactory bulbs. So that's right here. So this is on the ventral side of the brain. And so the end result, what we're trying to do, obviously it's for smell, right? So there's all these different compounds that are in the air, and they need to be volatile. They need to be in the gaseous state in order for us to smell them, okay? So once we activate the receptors here within the olfactory bulb, we're going to get into all this process, but what it eventually leads to, the particular regions of where we're processing this information. So one is here within the frontal cortex. So I have this information written on the board, and then also some of the structures that are found within the limbic system. And so we've already identified components of the limbic system, and so if I take this off, here within some of the deep structures of the brain, right, you can see the hippocampus, and then the amygdala is within this region as well, within the deep structures of the brain. Okay, so I'm going to work my way to the board. And so what I have drawn over here, this is, so I also have the head model here. So the olfactory bulb, it sits in this particular region where the cribriform plate is. So when you activate it, there's all this like mucosa that's here at this particular region at the head model. And so that's what is depicting here. So number 1, this is what's indicating the olfactory bulb, which is what I just pointed out. And then this particular region where that's in green, that would be the cribriform plate. This is connective tissue that's connecting the epithelium right here. And then within this epithelium, the olfactory epithelium, this is where all of these olfactory sensory neurons are found. So in order to activate the neurons themselves, the compounds, the volatile compounds, they'll bind here within the olfactory cilia. So that's these little projections that are coming off. So then if we work our way this way, let me stand on this side. So here at this olfactory cilia, that's where we have this phospholipid bilayer. And so in order to get this signal to the brain, to get what we were talking about here earlier, we have to activate what's known as a secondary messenger system. And so when the odorant molecule binds at its receptor, right here within the olfactory cilia, it activates the secondary messenger system. So what is a secondary messenger system? So what we're trying to get to is the point where we can activate a sodium channel. Because what happens whenever a positive thing flows into the cell? What happens to membrane potential? It becomes more positive. It goes up. So that's what's going to send this action potential all the way along this particular sensory neuron, and then we activate these. It'll activate the next neuron and then we'll continue this cascade until we get to the particular regions in the brain. So what the secondary messenger system is, so once the molecule binds to its receptor, we have what's known as a G protein. So it's just a particular name. It has a guanosine residue. And so once this receptor is activated, one of the subunits comes off. So that's the G protein in the alpha subunit. So once that occurs, it activates an enzyme that's known as adenylate cyclase. And then that's where we get to our actual secondary messenger. So our secondary messenger is what's known as cyclic AMP. So the reason we're able to get this is because adenylate cyclase converts ATP into cyclic AMP. And then the cyclic AMP is what will activate this sodium channel to allow the ion to flow through, and therefore we can propagate the action potential and then get it to the brain. Okay, so are there any questions regarding olfaction and smell? No. Okay, so the next thing that we're going over is gustation. And so obviously, so this particular region right here, this is where the tongue is. And so at the back of the tongue is where we have this particular structure, which is known as the valate papillae. So at the valate papillae, I'm blowing up this particular structure, that valate papillae, and that's what's shown here. And then what's shown at this particular region is just that part that's blown up. Okay, because once again, where we're trying to get to, the whole point of this is that you have to activate the receptors that are found here within this gustatory epithelial cell, and then get it to the brain at the gustatory cortex. So the gustatory cortex is found within the insula, and the insula is what we identified earlier in the semester. So that's what this particular region here is on the lateral side. So that's where we're trying to get to. So how does this occur? So we have different types of taste bud receptors. So sour, salty, sweet, umami, and bitter. And depending on which ones you activate will determine how the signal is processed and transmitted. So for sour and salty, it's for basically acidic foods, and then also sodium, so salty foods. So this will activate ion channels where they flow in and the action potential propagates, compared to these particular ones in which we activate the secondary messenger system. So once the compound binds to its receptor, so this is where the taste port is, it'll then activate the so for instance, if it was like a secondary messenger system, that's what's occurring within this specific type of cell, the epithelial tissue, right? Because these are all a bunch of cells. And then from there, we'll finally get to the point where this action potential is propagated along the taste fibers of the cranial nerve. So then the action potential will be propagated, and then these are the cranial nerves that are responsible for taste. So cranial nerve 7, which is facial, 9, which is glossopharyngeal, and then from there, right, there's synapsing, just like what we talked about earlier as far as like the ascending tracts. Remember, it's a similar thing here, like first order, second order, and third order and everything. But it goes, there's a synapse in the medulla, a synapse in the thalamus, and the final synapse is within the insula. Okay, so are there any questions regarding taste? No? Okay, so that's going to do it for this lecture.